Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar, titled Measuring Key Analytical Attributes of Viral Vectors with Macromass Photometry. I'm Abby Pinchbeck, an editor at BioInsights, and joining me today is Laura Palla, who will discuss how macromass photometry enables fast, accurate, label-free analysis of viral vectors, improving characterization and streamlining cell and gene therapy production. Now, I'd like to introduce our presenter. Laura Palla is a product marketing manager at Refine, with extensive experience in product development and management of scientific instruments in nanomedicine and gene therapy, holding a PhD in organic medicinal chemistry focused on targeted drug delivery systems. So, without any further ado, I'm going to hand over to Laura to kick us off with her presentation today.
Thank you very much for the introduction. I'm really excited today to be here to tell you more about macromass photometry and how it can be used to assess key analytical attributes of viral vectors. A little bit about Refine. We are a global company with presence in United States, Europe, as well as the APAC region. I would like also to tell you more about the current solutions we have on the market. We have the 2MP product line, including the manual and automated version, which allows for single molecule mass measurements for a mass range between 30 kilodaltons and 5 megadaltons. We have also the SamUX-MP product line, including, the manual and automated version. These instruments were developed to characterize AAVs for the gene therapy space, and they have available also a GMP-compliant software. And the last one here is the KaritroMP, which allows for characterization of larger viral vectors compared to AAV, and specifically particles between 40 and 150 nanometers using an automated workflow. And this is the one that we are going to be talking about today. KaritroMP is a new tool for rapid analysis of large viral vectors. It's a benchtop instrument that allows for assessment of purity and stability of adenoviruses and lentiviruses within minutes. But what are the challenges that we would like to solve? So starting with adenoviruses. While producing adenoviruses, you would have, in addition to your desired product, so the full functional adenovirus, a series of impurities ranging from empty adenovirus, the helper virus, which sometimes is used during manufacturing of the adenoviruses, as well as fragments and degraded capsids. These impurities should be assessed as well as removed because they either decrease drug efficacy or they pose safety issues. There are analytics out there to characterize adenovirus species. However, they are either non-scalable, such as analytical ultracentrifugation or electron microscopy. They are unable to quantify impurities, such as Titer assays, or they cannot really resolve populations, such as physicochemical techniques to measure size. Therefore, the need is to have some fast in-process analytics able to quantify adenovirus populations. But why is it important to characterize adenovirus during the process? Well, if we look at the process itself, you will see that it is composed by multiple steps in both the upstream and the downstream purification, and overall it takes several days, if not weeks. It would be ideal not to wait until the end of the process to see the purity and the quality of the product, but rather to monitor each and every step, because in this way you would be saving time and resources. So how can Karitro help with this? So Karitro is a single particle technique. It can resolve and quantify adenovirus populations, such as empty and full population. It's an automated walk-away analysis and is fast and easy. The technology that the KaritroMP uses is called macromass photometry, and it measures two parameters at once: the scattering contrast, which depends on mass, and the particle height, which is a measurement of particle size. Let's look at how it works. We have a measurement at a glass-liquid interface where we have a glass slide on top of which we deposit a droplet of sample containing the particles. The viral particles would land on the surface, taking advantage of non-specific binding, and what we would do is send a light source from the bottom, illuminating the samples. Each and every particle would scatter light, and this light would be interfering with the light reflected from the glass surface. This interference between the scattered and reflected light, which is also called scattering contrast, depends on both mass and refractive index, and therefore it could be treated as a proxy for mass or non-calibrated mass axis for single particles. In addition to this measurement, we also collect the particle height as a measurement of particle size. We do that by sweeping through focus and collecting the vertical dimension of the particle on the glass surface. Let's look at the contrast measurement more in detail. Macromass photometry weighs particles with light. So each particle would produce a contrast signal, which is the interference between the scattered and reflected light. And the contrast would depend on the particle mass. So heavier particles would produce a larger contrast signal. For example, in here, we have the comparison between two particles of the same size, but one of them is empty and the other is full. The full particle would be heavier, and therefore its contrast signal would be larger compared to the empty. In this other example, we have particles that have the same mass, but different size. So we have a small full particles versus a large empty particles. They would appear at the same contrast value, but they can still be differentiated thanks to the size difference. As mentioned earlier, the KaritroMP can also measure particle height. Now let's look into these two different scenarios. When working with rigid particles, such as Adenovirus capsid, the particle height measured with the KaritroMP would correspond to the particle diameter measured in solution with other techniques. When, however, we are working with soft particles, such as lipid-based vectors, the particle height measured on the Karitro is lower compared to the particle diameter measured with other techniques. This is due to the intrinsic deformability of these soft particles, which would interact with the surface where we are measuring them. Therefore, in this case, specifically, the particle height is a measurement, an indication of the particle diameter, but also of the intrinsic deformability of the particle, which often depends on its structures and physicochemical properties. Let's see how the KaritroMP can differentiate populations. In this hypothetical scenario, where we are measuring size versus contrast, as we would do in a Karitro fashion, we have a small empty particle appearing at a certain size and contrast value. If we measure a full particle with the same size, this would appear at a larger contrast value, so larger mass because of the payload, but same size value because they have the same diameter. Let's imagine now to have a large empty particle with the same mass compared to the small full particle. This would appear at the same contrast value, but in increased size because of its larger diameter. If we have a large full particle, we would be able to differentiate it from the large empty because of the mass, even though they have the same size. So the value of Karitro here is its capability to differentiate particles which have the similar size but different mass, but also the capability to distinguish particles which have different size but similar mass. I would like now to show you how contrast can be used to resolve particles of similar size. And specifically here, what we are showing are the data of our size calibrant, which is called sizeference. This is an equimolar mixture of 5 populations of silica beads, ranging from 50 to 120 nanometers. When measured on the KaritroMP, what we can observe on the size axis is a broad histogram where the 5 populations cannot really be resolved from each other due to the fact that the size values are quite close, quite similar. However, if you look at the contrast axis, you can clearly see 5 peaks, and you are even able to resolve the 50 from the 60 nanometer beads populations. So in conclusion, the Karitro, thanks to the contrast axis, can resolve populations that have similar size, but taking advantage of their mass differences. Let's look at how the KaritroMP can help differentiate populations based on mass and size with adenovirus samples. So in this case, we have a mixture of fragmented capsids, empty and full capsids, and you can really tell them apart based on both axis information. So the fragments appear at low size and contrast together with protein aggregates, You can see a distinct peak on the size histogram as well as the contrast histogram. Then you can see another major size peak at around 80 nanometers. Underneath this peak, you can see the presence of two populations, your empty capsid and the full capsids, which have the same size but again different mass. These two peaks can be really easily distinguished and resolved on the contrast axis. So that's how Karitro can really help getting information on the various populations thanks to the two parameters measured. Now let's look into some more adenovirus KaritroMP data. As mentioned earlier, the Karitro can distinguish the various populations from fragments to empty and full adenoviruses, assessing effectively adenovirus purity. We assess the ratio between the empty and the full capsids, and here you can see from the standard deviation values over the triplicate measurements that this is quite reliable and robust. In order to investigate whether the Karitro could reliably quantify adenovirus empty to full ratio, we did this piece of work in collaboration with the Jenner Institute at the University of Oxford. They measured different empty to full mixtures ranging from 0% full to 100% full. So the two populations, empty and full, were mixed together in different ratios, and then the percentage ratio of the full and empty were measured on the KaritroMP. These data were then compared to the theoretical percentage calculated from the mixing ratios. And what you can see here is a very nice linear positive correlation between the measured and the theoretical values. You can see a very small offset between the raw values and the linear theoretical values. And this is coming from a small percentage of full adenoviruses into the empty sample. This is related to the fact that it was difficult to purify completely the full adenovirus from the empty sample. In conclusion, what we can see here is a very nice way to determine empty to full ratios with the KaritroMP in a few minutes with very small volumes of sample. But let's look at now, how does Karitro measure up? How does it compare with other technologies? So the KaritroMP is actually the fastest tool for robust empty-full quantification for adenovirus samples. So in this piece of work that the Jenner Institute's Oxford University performed, it was compared the empty to full ratio with the KaritroMP, which is the orange line, with other technologies, and specifically analytical ultracentrifugation, negative stain electron microscopy, and an HPLC method. What we can see in here is that actually the data collected on macromass photometry are the ones that are closer to the expected theoretical ratio. And on top of that, we need to point out that macromass photometry is also the fastest tool amongst those listed here, meaning that the performance of the Karitro for empty full determination is actually quite convenient versus other methods due to its low cost and high speed. The KaritroMP can be used to monitor adenovirus production processes. In this example, we are observing a comparison between a clarified sample, one of the elutions, and the drug substance. What we are seeing is an enrichment into the full population that goes up to 71% across the total particle measured with the Karitro at the end of the process. This means that the KaritroMP can be used as a convenient tool in process to monitor sample purity, because it requires minimum sample amounts and the measurement takes only a few minutes. Not only Karitro can differentiate empty and full capsids, but it can also differentiate the full adenovirus from the helper virus, which is sometimes used during its production. So the helper virus should be removed at the end of the process, being immunogenic, and therefore it needs to be assessed and then removed from the drug product. In the first graph here, we are showing the full adenovirus, which is the drug product, which appears at a certain contrast value and size value. The helper virus on the right appears at a larger contrast due to the fact that it has an increased mass, because its genome length encapsulated is slightly longer compared to the drug product. Therefore, what we can do with the Karitro is assessing and quantifying the presence of helper virus in final products. The KaritroMP can also monitor adenovirus stability. In this specific example, we are studying thermal degradation of an adenovirus sample, which was heated up at 47 degrees and measured over the course of an hour. You can see one main adenovirus populations at the 0 time point, accounting for 47% over the total. After 15 minutes, you can see that the adenovirus population decreased to about 18%, and we are the appearance of other populations at lower contrast, so lower mass, as well as lower size. At 60 minutes, so after 1 hour, what we observe is the increase of the presence of these additional new populations that are likely degradation product and the disappearing of the adenovirus population. To conclude, the Karitro can be used as a fast tool to monitor adenovirus stability and to study the presence of new emerging degradation product. Let's now look into lentiviruses. Which are the challenges when producing lentiviruses? When producing lentiviruses, together with the functional lentivirus particles, there are a series of impurities that should be quantified and ideally eliminated as well. We have a mixture of non-functional lentiviruses, which are essentially lentiviruses missing some components and therefore are not infectious, and also some exosomes or extracellular vesicles that again are not infectious and should be ideally removed. In terms of analytical tools available for lentiviruses, the gold standard is definitely the infectivity assay, which would allow to know how many functional particles are in there. However, this would take several days and is definitely not suitable for in-process measurement, but only can be used for the final product. The need here would be to have some fast in-process titer estimate that can be used before the end of the process and on the final product in order to assess whether the production is going in the right direction and if not, ideally stop it earlier. So why is it important to characterize lentivirus in process? Similar to the situation described earlier for adenoviruses, the production process for lentiviruses is long and composed by several steps, which overall take days, if not weeks. If you wait until the end of the process and only find out at the end that there was something not going right, effectively you wasted a lot of time. So if you could monitor each and every step and assess whether the production is going towards the right direction, it would save a lot of time and ideally resources as well. Let's look at how the KaritroMP can help. Karitro can give a physical titer estimate. It's an in-process automated tool, and it allows single particle analysis. Karitro can differentiate lentivirus populations with no sample prep. If we look at this example here of a lentivirus sample, we can see that this appears much more heterogeneous compared to the adenovirus shown earlier. However, by using a gating strategy, we can distinguish the various populations present in there due to their different values in both size and contrast, so size and mass, and specifically the lentivirus-like particles from the EV's extracellular vesicle particle, which appear at a much lower contrast due to their lower density values. But let's look more in detail into lentivirus Karitro data. As shown in the previous example, lentivirus samples tend to be quite heterogeneous, and therefore our question was how can we identify and distinguish lentiviruses from contaminants. In this example here, we are comparing lentivirus sample from the supernatant with two negative controls. We are observing the presence of a population in this gated region in the lentivirus sample on the left. However, this population is absent in the two negative controls, which helps us assessing where the lentivirus are to be expected in these Karitro graphs. In conclusion, the Karitro can distinguish lentiviruses from impurities and specifically lentivirus particles from extracellular vesicles and protein aggregates. The KaritroMP can assess lentivirus purity in process. In this example here, we are comparing a crude and a purified aliquot from the same process. In the lentivirus region, we are counting the percentage of lentivirus particles over the total, and we are showing that they increase by tenfold from the crude to the purified, going from 2% to 24%. We compared this increase with the infectious titer, which shows a sevenfold increase, nicely matching with observed result on the KaritroMP. So the conclusion here is that the KaritroMP can be used for a fast process monitoring to determine the sample quality before performing infectivity assays and having a quick overview of the sample purity. And of course, the next question was, can we determine a correlation with titer? And the answer is yes, we can. We demonstrated, in collaboration with Professor Dirk Lindemann's lab from Dresden University, how the Karitro lentivirus region counts correlate positively with the physical titer that they determined by PERT assay, so virions per milliliters. Different sort of particles were measured, so we had infectious particles, non-infectious particles, as well as negative controls. And we observe a very nice positive correlation between the two methods, suggesting that the KaritroMP can be used for a fast titer, physical titer assessment in process in order to quickly take decisions on the next step in process. Let's now look at the consumables and workflow. The KaritroMP sample carrier is composed by three parts: a glass slide, a sample well cassette composed by 14 wells, allowing for the measurement of up to 13 samples plus one calibrant, and then a plastic lid. The lid allows for the safe handling of the sample carrier, which could be, for example, loaded under a biosafety hood and then moved safely, once closed, to the machine outside of the hood, limiting effectively the exposure for the operator to biohazardous samples or potential contamination issues. Another consumable is sizeference, which I introduced a few slides ago, which is a mixture of silica beads of different size that allows for the calibration of the particle height axis and also for the internal calibration and reference of the contrast measurement. Let's now look at the workflow. So the KaritroMP uses an automatic acquisition workflow. There are 20 minutes which are hands-on, involving the sample prep, the loading of the sample carrier, placing it on the instrument, and setting up the software. And this part is followed by an hour and a half fully automated part, which involves the measurement of all the 13 samples plus calibrant sequentially and the production of the results on the fly, which will be ready at the end of the run to be exported by the operator. Macro mass photometry is quite an advantageous technique. It involves limited sample preparation, pretty much only dilution to the appropriate concentration. It's fast, taking a few minutes per sample, and automated, so for the full run of the cassette of 13 samples it takes about 2 hours, including sample prep. It requires only low sample amount. We are talking about 5 microliters per sample at a concentration of 10 to the 8, 10 to the 9 particles per milliliter. It's easy to use. Training internal people as well as customer takes about a day. It has a small footprint, which is quite convenient in a lab space, and the operational costs are below $5 per sample. Karitro is able to bridge the analytical gap between techniques which are fast but poorly informative and techniques which are critical, very informative, but very time-consuming as well. The Karitro value is that being so fast, it can be used during the process to monitor how this is going, and it can be used to take further decisions on the process itself or triaging the samples for further analytics such as infectivity assay, effectively saving time and resources. From the instrument specs, we can see that the size range is between 40 and 150 nanometers. The sample optimal concentration goes from 10 to the 8 to 10 to the 9 particles per milliliter. The sample volume required is 5 microliters, so quite low. As mentioned, the sample carrier capacity is 14 wells, allowing for 13 samples measurements. Time to result is 6 minutes per sample. Here you have the rest of the specs for what concerns resolution and precision, as well as the laser wavelength at 450 nanometers, the instrument dimension, and its weight. So in summary, the KaritroMP can assess lentivirus and adenovirus purity and stability within minutes. It allows for fast in-process characterization, achieving new information on populations that help assessing purity and stability early. And it can also assess faults during the process quite early in order to take a fast decisions on the next step. Before concluding, I would like to show you some preliminary data collected on lipid nanoparticles. While the Caritro was launched on the market for the viral vector space, we currently are exploring into different application spaces, specifically lipid nanoparticles. So we are developing a solution which is not yet commercially available, but I would be really happy to show you some preliminary data on the lipid nanoparticles. In this example, we are comparing empty and mRNA-loaded LNPs on the Caritro. You can see on the contour plot here the overlay of the contour line of the empty in blue and loaded LNPs in orange. You can see that there is a shift towards larger contrast values, but this is even more clear on the 1D contrast plot, where we definitely can see a shift towards larger masses with the loaded LNP. And you can see the numerical value in these bars on the bottom, where we have the median value on the top of the bar, and the IQR, so the interquartile range, which would correspond to the broadness of this peak, or to the heterogeneity, let's say, written on the right of the bar. So what we can see here is that upon loading, the LNPs shift at the values of larger mass, which is to be expected. The next step was to compare lipid nanoparticles with different mRNA payload. In here, we are comparing an empty LNP with a mRNA-loaded LNP with a low payload and another one with a higher payload. The contour plot here shows a very different heterogeneity of the three samples, with the gray being the one with the high payload, being the most heterogeneous of the three. From the 1D contrast plot, you can observe a shift towards larger masses as the payload increases, but also an increase in heterogeneity on the contrast axis, which tells you that the mass distribution is more heterogeneous as you increase the payload. In this other example, we are comparing three different formulations. We know from the partner that the gray sample, so P2F2, is coming from their optimized process. So they were actually quite happy to see that this also appears to be the most homogeneous on the contrast axis. We can see that from the lowest IQR value, so this bar here, and the size values that we measured are actually quite comparable to each other. So again, the value that Caritro provides here is by looking at heterogeneity on the basis of mass, which can be sometimes very different from the size measurement. In this other example, we are showing two different formulations containing the same lipid formulation, the same N-to-P ratio, but different RNA type encapsulated. So the blue sample contains mRNA, while the orange sample contains siRNA. What we can easily observe is a very different heterogeneity between those two, with the blue one, so the mRNA-loaded LNPs, appearing much more heterogeneous on the basis of mass. This is clear from both the contour plot but also the 1D contrast plot, where you can see that the tailing of the mRNA-loaded sample is much broader compared to the siRNA-loaded sample. This is probably due to the fact that siRNA is a much smaller molecule, and the way it packs inside of the lipid nanoparticles is different compared to mRNA, which is a much larger molecule. In this degradation study, we are observing the effect of keeping the LNP sample, mRNA-loaded sample, at 4 degrees or at room temperature after 8 days. As you can see, after 8 days at 4 degrees, the sample appears pretty much the same. But at room temperature, you have the loss of the major LMP population, effectively showing degradation. What I showed you so far were some really exciting preliminary data collected on lipid nanoparticles. We currently have in place an early adopter program for LMPs, and I would like to invite you to get in touch with us if you are interested in learning more about that or in taking part. We would be really excited to have a chat with you and move this forward. And with that, I would like to thank you all for your attention, and I'm happy to take your questions. Thanks very much.